So this is our talk sponsored by Thermo Fisher Scientific, and the presentation will be by Dr. Kamila Schmidt from Manchester University. So as we're short on time, I'm going to hand this over now to Kamila to walk through the presentation.
Offline breath analysis, storing up trouble. The analysis of VOX is unique in its ability to quickly and reliably detect both normal and abnormal processes in the human body. The analysis of VOX in the exhaled breath is particularly notable because of its non-invasive nature, the fact that it allows for frequent and rapid sampling, and that the samples are easy to collect. GCMS is considered the gold standard for identifying compounds in breath analysis because it provides detailed mass spectra and high chemical specificity. However, GCMS is an off-site method. Typically, in breath analysis, we firstly collect breath samples. Then usually the samples require transporting into the lab, where sample preparation and analysis takes place. After the sample preparation, the samples are usually stored for some time before analysis. Therefore, preserving the integrity of breath samples during transport and storage is one of the main challenges in breath gas analysis. Storage temperature, humidity, time of storage, as well as adsorbent type of thermal desorption tube will have an impact on various physical and chemical processes that can affect sample quality and integrity. These processes are compound degradation, contamination from pollutants, retention in containers, and interactions among sample constituents. They all can lead to irreversible changes, distorting the final chemical analysis results. Previous studies of thermal desorption tubes storage conditions give a mixed picture. For example, this study found all 15 compounds but one to be stable during the first 4 weeks of storage. One compound was found to be stable for 90 days, and no storage temperature analysis was done here as all the samples were kept at -80. This study showed differences in VOC recoveries depending on adsorbent type, temperature, and concentration of the standard. Refrigerated temperatures were not advisable here for Tinax TA adsorbent type. Yet another study found no consistent differences in recoveries for the different loading or storage conditions, and also they found no difference for recoveries between tubes stored in the fridge and at room temperature. Two mini studies presented today are part of a clinical study of VOC biomarkers of asthma and pet allergies. When it comes to thermal desorption tube storage duration, it was an opportunistic study. We moved labs, and it took time for some breath samples to be analyzed. We were able to analyze storage periods of up to 10 months using a small set of standard samples here. The second study, investigating storage temperature, was using breath samples. Methods for our standard samples are presented here. Standard samples were prepared by injecting standards in methanol using Mark's calibration solution loading rig on Tinax GR tubes. We included VOX previously investigated in our asthma studies as standards. The samples were next analyzed on TD-GCMS with audit trap detection, on Q-Executive by Thermo, in splitless mode, using full scan between 40 and 350 mass to charge ratio, and in positive ionization mode. The data was processed with Chromium for peak detection and integration, and statistics was performed in MetaboAnalyst. The analysis for long-term storage of thermal desorption tubes was conducted for 21 compounds using a small set of standard samples. The samples were stored for various amounts of time. They were stored overnight, they were stored for 4 months, for 5 months, and for over 10 months. Some compounds appeared to have significant level differences between the analyzed times. For others, there was no change in levels over this period of time. On the PCA plot, we can see a separation of standard tubes stored for over 10 months, but no real separation for the other times. Patterns for the Vogues were different. Here are a few examples. Isoprene appeared to be at higher levels in overnight tubes and tubes stored for 5 months, and lower levels for 4 months and over 10 months tubes. Levels of nonane were similar for 4 and 5 months storage, lower for overnight tubes and over 10 months tubes. Levels of decane were the highest in 4-month tubes, but there was no difference for the rest. Pyridine showed the highest levels for 5 months, and tridecane showed the highest levels for overnight tubes. Again, lower levels appeared for over 10 months tubes for both compounds. There was no difference for some compounds stored for 10 months at all, for example, limonene. We can state that there is a significant difference in Vogue levels for tubes stored for different times, depending on the compound. Breath samples were collected using the receiver breath sampler. Volunteers were asked to breathe normally for around 10 minutes. We collected approximately 500 milliliters of lower airway portion. The samples were dry purged for 4 minutes after the collection. The breath collection was again performed on Tinax GR tubes. Samples were analyzed on Q-Exactive TD-GC-MS and again processed with Chromium, and MetaboAnalyst was used for statistics. Our second analysis asked the question whether there is any difference in Vogue levels for tubes stored at 4 degrees when compared to tubes stored at room temperature. The analysis was done for breath samples, which were collected during the same breath test, stored for the same duration, and analyzed at the same time. That's why we went for a paired t-test analysis. The analysis was done for 13 Vogues, and these 13 Vogues showed significant differences between the two analyzed storage temperatures. The PCA shows a separation between a red group, so tubes stored at room temperature, and green refrigerated samples. Here are the box plots showing the direction of the change. For all the analyzed compounds, the levels were found higher in the tube stored at 4 degrees. These are marked green in the box plots. So all the analyzed compounds showed a loss of Vogues from the tubes kept at room temperature, marked in red. There is a significant difference in Vogue levels for tubes stored at 4 degrees when compared to tubes stored at room temperature. To conclude, this study has added further evidence to the fact that the storage recovery of Vogues can be greatly affected by time and temperature. Our study showed that different Vogues show different time storage patterns. Some compound levels seem to get lower with time, some didn't show any difference. There were compounds that seemed to increase in levels over time as well. The study also demonstrated that storage temperature has a huge impact on the stability of the same compounds. The amounts detected were lower when tubes were stored at ambient temperature. Finally, evaluation of storage stability for each compound of interest should be a part of every breath study. At the end, I want to thank people from the research group I belong to. Firstly, Steve Fowler, Professor of Respiratory Medicine at the University of Manchester, and then Dr. Waqar Ahmed, a research fellow at the University of Manchester, and Dr. Ran Wang, an academic clinical lecturer from the University of Manchester, all heavily involved in work in our Rheomics group. And finally, Sam Sadiq, a CMS application specialist from Thermo, for her patience in helping me with Chromium. Thank you very much.
Thank you, Kamila. If you could please show your video.
That's interesting, isn't it?
I see your background, but not you. But
exactly. That even... Oh, there
we go. Yeah. If you just get a bit closer, maybe. There we go. Perfect. Thank you for a very nice talk there. Just a few questions here. In terms of the storage conditions itself, what do you have any sort of general recommendations from the work you've done for a discovery, sort of untargeted discovery study versus where you may be sort of looking at in a more targeted way at specific compounds?
Yeah, definitely. So for discovery study, because it's an untargeted analysis, and obviously we don't know what compound we will be analyzing, we basically, the recommendation is the storage to be as short as possible, or as short as possible in practice, because of course different projects will have different demands when it comes to logistics. But this is all to avoid any loss of our compounds of interest, any avoiding contamination, avoiding rearrangements, and so on. Literature studies varies between a week and 4 weeks. So that's kind of recommendations. Everything changes when it comes to targeted analysis, of course, here. Of course, if you've got standards, you can just test the stability of your compounds, and that's that.
Yeah, thank you. You looked at, I think, 4 degrees, and have you looked at -20 or other temperatures sort of in between?
Yes,
I think you looked at
that's
-80 as well.
Question. That is an obvious question, isn't it? So as far as I'm aware, there hasn't been any study yet done when it comes to comparison of -20, -80 versus the fridge storage conditions for breath samples on thermal absorption tubes. So yeah, depends on the lab. Some labs store the thermal absorption tubes at -80, others like ours store them at the fridge, and at this moment we cannot really state what works better or what is recommended.
Yeah, thank you. And then I think an important question here, and something we do at Owlstone, did you dry purge the tubes before the storage period?
Yes, we do tend to dry purge our samples as soon as possible after the sample collection.
Yeah. Okay. And just in terms of the standards you're using, why did you choose those particular chemicals for this work?
Yes, so it is a standard, a mix of polar and non-polar compounds, representative compounds found in breath, but not only. It's also straight chain alkanes, ladder. As you could see in our study, we didn't include all of them in the analysis. Some weren't detected, others were below the limits of detection, especially for the breath samples. Some were also not coeluting, so we couldn't include them in the analysis as well. So.
Okay, thank you. All right, we better leave it there so that we can break for lunch.
Yeah,
Thanks very much again, Kamila.
Quite hungry. Yes.
Yeah, absolutely. Thank you very much.